Hi, I'm Rafael Canevarolo, a postdoc at Moffitt, and my work is called Dynamic Epigenetic Landscapes Define Multiple Myeloma Progression and Drug Resistance. In our work that's going to be presented in ASH 2020, we studied multiple myeloma, which is a plasma cell cancer. We don't know yet exactly what are the causes that drives disease onset and drug resistance. And the problem is because of that, multiple myeloma is still an incurable disease. By studying a very large cohort of patients that were treated at Moffitt, more than 1,000 patients, we are able to see different patterns in gene expression of those cells. And we saw that, for example, pathways related to cell identity were decreased whilst during myeloma onset, while pathways related to metabolism and cell cycle were increased during drug resistance. And later on, we tried to find what could be driving this. And we found that there were some epigenetic markers in histones that wraps the DNA. And these markers were differentially concentrated during disease stages. We confirmed that in 10 myeloma samples with different disease stages using a technique called single cell ATAC-seq. And later on, we are able to map those changes to very specific super enhancer master transcription factors related to plasma cell identity. And we saw that some of them were decreased during myeloma onset, and many other ones were increased during drug resistance. So we came up with this bow tie model in which we could map a lot of mutations and cytogenetic abnormalities with this reprogramming of what we call the epigenome, leading thus to an increase or decrease of these biological pathways that I mentioned related to myeloma onset and with drug resistance.